Time for acute infections. Yeah, that's good. All right, you've heard about persistent infections from Dr. Silverstein, so now we're going to talk about infections by viruses that are really quick, or relatively quick, acute infections. And if we look at the way viruses replicate in cells, we can get some clues about the basis for acute infection. So if you remember, some virus infections kill cells in culture. You infect a plate of cells, the virus replicates, it kills them, make a lot of viruses, and that we call cytopathic virus. Okay? Then we have some infections that proceed without killing the cells, and these are non-cytopathic infections. These are, again, in cell culture that I'm talking about here. And then we have infections which don't kill the cell and they don't make any viruses. Maybe the viruses become latent right away, or there's some other reason why progeny are not produced. So those basic results in a cell culture can predict what happens in a host. And the host, of course, adds a second complicating factor, which are host defenses. So together, the pattern of the infection in a cell culture together with host defenses determine what's going to happen. So you've seen this before last week, I'm sure. And these are the different kinds or patterns of infection that we talk about. Today we're going to look at acute infection. So in this image, again, we have virus production in the blue line. And the disease is shown by the red area. So last time you heard about infections that go on for a long period of time. We call them persistent infections. There are all kinds of persistent infections where you have virus produced for a long time in the absence of disease, or intermittent disease. Today we're going to talk about things that are over usually in a week or two. You have a burst of virus production, disease, and then if you're lucky, everything is resolved and you recover. Acute infection. So these are all pretty brief. In a healthy individual, the virus is produced and eventually eliminated by your immune system within days. Some exceptions. There's some variability in timing, of course, but for the most part, we call them quick, and that's why they're called acute infections. And usually you just get one infection, if you're lucky. So we can make vaccines against these viruses. Do you know what an exception to this might be? What's an acute viral infection where you keep getting infected over and over again? You have to know this. A rhinovirus would be one. Any others? Influenza, right. So those are special cases where the virus changes. In the case of influenza, or in the case of rhino, there are so many of them around that you can never be possibly infected with all of them. As you know, some acute infections can kill you. Even influenza, which can be just a routine infection, kills 10,000, 15,000 people or more every year in the U.S. And even in healthy people, apparently healthy, that can happen. So obviously there's some underlying issue that we don't know about. The acute infection that kills you is also characteristic of zoonotic infections, where the virus comes from an animal and jumps into a person. It's not well adapted to the person, so it wipes them out. There's not a good balance between the virus and the host. But for the most part, we can handle acute infections. So here's another slide you have seen, and it is the course of a typical acute infection. And the red line, of course, is virus production with time on the x-axis. So here the virus comes into you. And the innate defenses are alerted, of course, and the virus begins to replicate. And if the innate defenses do not handle the infection, the amount of virus increases with time. And after a certain point, there is a threshold where the adaptive immune response is activated. So that's shown as this blue bar here. So eventually the adaptive response will be activated, but that takes time, so the virus is continuing to replicate. And eventually, the adaptive response will clear the infection, but we've had some disease already because the innate response couldn't handle it. The virus eventually is cleared, you recover, and then you have the memory of the infection, which hopefully will prevent you from getting infected again, or at least make the infection milder. Again, with exceptions, flu and rhinoviruses being some of them. So before we go on and talk about some aspects of acute infections, just want to talk about trying to relate how viruses replicate versus the kind of pattern of infection they produce. So they're generally thought to be two kinds of viral replication or viral growth strategies. Okay, they're shown by these two graphs. The one on the left is the R strategy, and this is a case of exponential virus growth. We're looking at virus production with time. And this is the case where the virus has plenty of hosts to infect. A lot of virus is produced in each host, and it spreads readily from one host to the next. So the replication can continue indefinitely as long as there are hosts present. It's not limited by production or by the number of hosts. The slope of this can vary, of course, depending on the virus, but it's a straight line. Then we have the one on the right, which is called a K strategy, and this is characterized by typically lower virus yields. The virus may replicate very slowly. It may have very few hosts to infect, so this spread or the replication of this virus is limited by the number of hosts, which is K, essentially. And you see at some point this virus replication is limited because there are no more hosts present. So these are two distinct kinds of viral growth strategies which really correspond to acute and persistent infections in a general way. So the R replication strategy, as I said, large numbers of progeny maintained in the population by infecting one person after another, so there's not a limitation in the number of hosts. And as long as there are susceptible hosts, the virus can grow. And then the K replication strategy, where the virus doesn't replicate very quickly, and the new hosts may be rare or non-existent. And if you think about epidemics of virus infections, you can see how these work. So for example, influenza is an R replication virus, but eventually it runs out of susceptible hosts because there's a lot of immunity in the population. So this last point, no limit as long as there's susceptible hosts, no longer holds. So R replication strategies, as you might predict, are manifested in populations as acute infections. So they have short reproductive cycles, they make a lot of progeny. Often the disease is not obvious. We're going to talk a lot of examples where there are inapparent infections, that is, there's no apparent disease. And this is another characteristic of an acute infection. There are high rates of shedding, virus transmission. And again, if you don't have enough susceptible hosts, either numbers or because of immunity problems, the infection burns out. And you can mimic these kinds of situations in cell culture by infecting at a low multiplicity of infection. You put very small numbers of viruses into a cell culture dish, and it has to go through many rounds of replication to infect the entire dish. It makes a lot of virus, it replicates quickly, but when the cells are exhausted, when there are no more susceptible cells, the infection stops. So that's a typical R replication or acute replication strategy. The K replication is typical of persistent or latent infections, which you heard about last time. Very small amounts of virus are typically produced over extended periods of time, and the host typically survive for long periods as well. Now, there's a term we have to address. You might have heard it before. It's incubation period. And this is the initial period before the symptoms are obvious. The virus is in you replicating, but you don't display any overt symptoms. As soon as you have something measurable, a fever, a rash, coughing, then the incubation period is over. And so viral genomes are replicating during the incubation period. The host is responding, and as soon as enough cytokines are made, remember, you're going to start having these global symptoms of infection, the fever and muscle aches and so forth. Fever, malaise, aches, pains, and nausea. The incubation period is quite variable, and in the end, it's going to be a matter of how we classify infections. It can be as short as a day or two. In the case of AIDS, it can be years, although that's probably not an acute viral infection any longer by definition. So a short incubation period, a couple of days or maybe a week at the most, or 2 weeks, usually means that the virus multiplying at the initial site of entry is what is producing the symptoms. So influenza enters your respiratory tract, begins to multiply in the tract. You have very short interval between when the virus comes in and you start having symptoms of influenza-like illness. The longer incubation periods are a consequence of actions beyond the primary site. So measles has a longer incubation period than influenza, the time before appearance of the rash, because the virus has to spread from the respiratory tract through the blood to the skin. So a longer incubation period means the virus just has to go somewhere else. And that's measles. So here are some incubation periods of common viral infections. Again, these are not all acute infections by our definition. The shortest ones for influenza, rhinovirus, or other viruses that cause common colds, other respiratory diseases. So the respiratory diseases are the shortest because the virus enters where it's going to cause the disease. Then when you have infections where the disease is more distant from the entry point, the incubation periods get longer. Now we start to move into the many, many months of incubation period down here. Let's take a look at rabies. 30 to 100-day incubation period. So, you know, I would still classify rabies as an acute infection because the virus comes in, replicates, it causes disease, and then you're dead in this case, unfortunately, because rabies is almost 100% fatal. This just takes a long time for the virus to go from where the dog or the bat bites you, has to spread up your nerves to get into your central nervous system, and that takes many, many days. So I think rabies is an acute infection, just has an abnormally long incubation period. AIDS, of course, has the longest incubation period on this table, 1 to 10 years. And you could look at AIDS, and we'll talk about AIDS on Wednesday, as an acute infection. Initially, there is an acute phase of AIDS infection when you have very typical symptoms, and then the symptoms go away and the virus incubates in you for 1 to 10 years. So you have an acute infection, but something goes wrong and you can't clear it, and it becomes a persistent infection. And that's why the incubation period is so long. So there are many inapparent acute infections. We've been talking about these on and off throughout the course. That is an infection where virus is replicating, but there are no symptoms. And these are successful infections as far as the virus goes because it's replicated and it's probably spread to another host, but there's no symptoms of disease. So you can make enough virus to spread in the population, but apparently not enough to produce symptoms. How do you detect inapparent infections? Well, in the old days, you could detect antibody rise in the individual. So if you were lucky to have a serum sample taken before infection and during disease or afterwards, you could demonstrate a rise in antibodies to the virus. Nowadays, you can use molecular methods to detect it as well. So if you suspect that there's an infection in a population, you can take samples of sera and look for the virus using these methods. Well-adapted pathogens often follow this example. Influenza virus, polio virus, even measles are well adapted to humans. They've been replicating in us for many, many years. And that's a typical pattern of not causing disease in everyone. A really good example is polio. Over 90% of polio infections are inapparent. So the ones who get paralyzed from polio are actually the minority. They're actually 1%. There's about 9% where you have mild symptoms, typical fever, aches, and so forth. And 1% you have the paralyzing disease. But everyone sheds virus. So you can see this is a significant means of spreading virus in a population. If you have people who are not showing any symptoms, they're walking around and spreading infection. And polio, when it started to cause epidemics in the 20th century, at the beginning of the 20th century, people were quarantined who were paralyzed, but 99% of the individuals who were infected were still walking around and spreading the disease. So that's why inapparent infections are common public health problems. It's one of the several reasons why they are common public health problems, because they can spread the infection without being detected. So acute infections are most commonly associated with serious epidemics of disease that affect millions of people every year. Polio at one time affected millions, no longer. It's nearly eradicated, but influenza still does. And measles numbers has gone way down, but at one time it was millions of people a year. They are often associated, remember, with emerging infections. Viruses that come from animals into humans have a particular vicious cycle because, as I said, the viruses aren't well adapted. But these are acute infections. And it's difficult for a number of people to deal with, from physicians to epidemiologists, drug companies, public health officials. Each have their own sets of issues to deal with acute infections. So why is that? Well, by the time you feel ill from an acute infection or mount a detectable immune response, most acute infections are done. All right? So think about that. If you have the way you detect an infection is by you getting ill or looking for antibodies, then it's too late to do anything about the infection. The virus has already spread on, so you've been shedding virus in this period when you're not showing any symptoms, so you're infecting others, and it's very difficult to diagnose the infection. In a large population, these viruses spread rapidly and explosively because of this fact, because they're inapparent infections that spread the virus in the absence of symptoms. So typical populations that spread virus infections very effectively are daycare centers, which are a modern development. So at one point there were no daycare centers, but there are always military boot camps, college dorms, nursing homes, schools, and offices. And there, a person will come in with no signs of infection, infect everybody else, and everyone gets sick before you even know that anything is going on. So that's the problem here. Explosive spread is possible in these conditions. And this is one of the reasons why we don't have a lot of antiviral drugs for acute virus infections, because by the time you diagnose them, the infection is over pretty much. So a drug is only useful in an acute infection if you give it very early on. So for example, Tamiflu, which you can have prescribed if you are diagnosed with influenza, you have to take it within 2 days of showing symptoms. Otherwise, it's a waste of time to use it. It will do nothing for you. And then that's a very difficult thing to do. So you have influenza-like illness, you go to a physician. It may not be influenza, but they'll prescribe Tamiflu anyway, and then you have the problem is that you have increasing drug resistance as a result. So one of the things that I think will change the landscape of treating acute infections will be to have rapid diagnostics. And so the one acute infection that we have some diagnostics for right now is influenza. But they're really lousy diagnostics. They're about 50% accurate. So we need better ones. These are the ones that you can have performed in the physician's office. I was talking to an emergency room physician at Columbia about this. He said they're about 50% wrong. So he says if someone comes in with influenza-like illness, you do this rapid test. If it's negative, then you do a PCR, which takes a couple of days. And then you're beyond the Tamiflu window, so you start them on Tamiflu anyway. So it's a real problem. And then what's going to happen in 5 years is really quick and accurate, rapid diagnostics. So how do we defend? Yes?
With the rapid diagnostics for flu, is it false positive or false negative?
They're false negatives. About half of the results are false negatives. Yeah, and then, well, in the end, most of the time you get Tamiflu anyway, no matter what. And then that contributes to selecting for drug resistance. So we need better diagnostics that don't take a couple of days. How do you defend against the acute infection? The innate response is extremely important for preventing acute infections, because as you know, the adaptive responses don't kick in for a couple of days. So as soon as that virus first comes in you, the innate response is crucial for clearing it. The innate response doesn't do everything, as you know. The adaptive response, in the end, will be crucial for clearing the infection. You cannot get rid of the infection with just an innate response. And in fact, here's an interesting question. What would happen if you had a strong innate defense but no adaptive defense? And the answer is that without innate defenses, the infection will spread and overwhelm the host. But the containment of the acute infection is essential. The adaptive defense is essential for that. So people without adaptive defenses, the immunocompromised individuals who don't have B cells or T cells, they have disastrous acute infections. They just can't clear them. Now, as you know from previous sessions, if you are infected with one of these viruses, you have memory. And this is an infection that usually happens only one time, like smallpox or measles or polio virus. You have memory and you don't get infected again. So vaccines can provide immune protection against those diseases. We'll talk about those separately later on. As we mentioned already, for some viruses, antigenic variation can get around the defenses and defeat the vaccine. So we need a new flu vaccine every year. And in fact, there is no vaccine against rhinoviruses. There are 100+ serotypes of rhinovirus, and no one's figured out how to put 100 serotypes in one vaccine. We have 20 or 30 serotypes of bacterial antigens in vaccines, but not 100. So the rest of the time I want to highlight some typical acute infections, go through the ones I like, and tell you what I know about them. Just as examples, we'll look at influenza and poliovirus, measles virus, rotavirus, and all of those are spread by human-to-human contact. And then we'll look at a vector